that look something like this. They have a body, they have a beating flagellum, such as many other swimming microorganisms, and then they have this collar of microvilli surrounding the flagellum. They're pushers, they swim around that way, and the sort of idea of the collar is that they will push out liquid by the beating flagellum, and that will draw in fluid through the collar, and then prey can get caught on the microvilli from where it can get back on cytosis.
So these are the blue stuff, you mean?
The blue stuff, yeah. That sort of.
But these ones do not move.
They don't move. They're a bit elastic, but in principle they don't move. Okay. The flagellum beats.
It's a single cell?
It's a single cell.
So what does it eat?
Smallest. It eats bacteria, small bacteria.
Okay.
So the scale here is around 5 microns, and the bacteria it eats are around 1 micron.
Okay.
So an important thing about it is where it lives in the tree of life. So we're all the way at the top. The black dot is the last common ancestor of the animals. The white dot is the last common unicellular ancestor. And then this M brevicollis is an example of a choanoflagellate. So and it's unicellular. So choanoflagellates are the closest unicellular relative that still is around. So this is one of the main reasons they're studied. Sort of this question of why and how it was that unicellular organisms evolved to become multicellular animals. Right. So let me just, there's going to be a bit of fluid mechanics in the beginning, but very little. You should all have seen the Navier-Stokes equations before. When you're very small and not swimming very fast, then the Reynolds number is very small, and that effectively means that you can ignore the left-hand side of the Navier-Stokes equation, and it becomes this much simpler and linear Stokes equation. Now this has a Green's function, so if you solve for a delta forcing, the solution is called the Stokelet and looks something like this. So obviously it diverges at 0, so it's not very nice for computational purposes. And when you want to do some computations, you can, for instance, use a regularized Stokelet that has a certain width to it, and that leads to what's called boundary element method. And I just wanted to introduce all this to show you how the flow field around a choanoflagellate looks like. So this is one that's stuck to a wall. So you can see that. So it's averaged around. So the flagellum has spun 2 pi, and then this is the average flow field. So you can really see how this beating flagellum drags in fluid through the collar, from where it then can eat.
Can I ask one? I'll ask a lot of things, but I apologize in advance. So this is not a numerical simulation per se. You just integrate. You use that integral that you showed us before.
I mean, I would call it a simulation, but.
But you have an exact formula. It's not that there is a grid or anything like that.
No, you don't have you don't have an exact solution. You have I've basically put regularized Stokelets on all these triangles and along all these splines, okay. And then the integral is done by a Gaussian quadrature, and then you have a linear system you can solve for the.
But there's no time stepping. That's probably.
Yeah. There's no time stepping. So that's for all Stokes flow. You don't really have. I mean, as soon as you define your forcing and your boundary conditions, the flow is defined everywhere. There's no inertial terms.
Okay. Perfect. Thank you.
But what I want to talk about really is colonies of these choanoflagellates, because they're very much studied because they are sort of borderline multicellular. What's interesting is that some of the species actually form colonies, and one choanoflagellate called Salpingoca rosetta forms colonies that look something like this, and you can understand why they get the name rosetta, because the colonies has this rosette-like shape, where the cells sit in the middle and the flagella point out in different directions like a flower. And under the microscope, stuck to a glass slide, it looks something like this. So this is sped up a lot. I think this was filmed at 1,000 frames per second. So the beats that you see there are around 50 beats per second. So this is how they look, but if I hadn't glued them to the glass, they would be swimming around.
So just one question on this, the previous part. So you kind of have a good idea of what the force distribution is that these flagellar beating are inducing on the fluid?
I mean, so there's all the, so from a theoretical point of view, yes, because you have tons of theory available, like slender body theory and similar types of theories that precisely, if you give a velocity distribution for a slender filament, you know the forcing.
Okay. That's what you use.
And that in terms gives you the flow. Yeah. So this, as rosetta exists in numerous types of morphology, so it does exist as single cells, a few different kinds. Some of them stick to the wall, some of them swim around slowly, some do fast. And then you can have two types of colonies. You can have chains, and you can have rosette colonies. And it's interesting, the rosette colonies only form if you have certain kind of bacteria in your solution. So basically you can have a monocenic culture that is with just a specific type of prey bacteria, and then you'll always form chains. But as soon as you add certain kind of bacteria, one called Algoiphagus, then they start forming these rosette colonies. And it's not known why one is advantage over the other. These are maybe organisms that if you do any kind of biological fluid mechanics that you're more familiar with, Chlamydomonas and Volvox. They are types of algae, and they have multiple flagella. So this Chlamydomonas has 2 flagella. This Volvox has something like 1,000 cells, and each with 2 flagella. And what's interesting about them is, and along with a lot of other organisms, is that their flagella are all synchronized. So you will see Chlamydomonas swimming around like this, or sometimes antiphase, but they'll be synchronized somehow. And the same in Volvox, you'll have synchronization within the 2 flagella in the single somatic cells, but you'll also have synchronization across the whole organism, and you'll have these mesochronal waves of flagella beating going down Volvox. So with this as inspiration, I sort of wanted to figure out, is this also the case in colonies of Choanoflagellates? So the way you typically study algae, or not typically, but at least in Ray Goldstein's group, is that you hold them on these micropipettes, and that works because algae has cell walls, so you can hold these on a micropipette and really neatly film the flagella, and even better is that the flagella beats in plane, so you can sort of rotate until they're beating in the plane and you can get really nice films. Unfortunately, that doesn't work with Choanoflagellates because they don't have cell walls. So the alternative is simply to get them to put some glue on the glass slides and look at them fall, and then hopefully sometimes you'll have them fall in the plane. For instance, here's an example. It's a bit hard to see, but you can see that we have indeed 3 beating flagella. It's a pretty bad movie. Sorry about that. So to track these kind of flagella, they sort of beat a bit in and out of focus, so you have to be a bit careful about the actual tracking. So what I ended up doing was defining this flagella tracking problem as an energy minimization problem. So you minimize sort of a snake energy, which is which punishes very high curvatures or stretches in the flagella. So we're sort of saying it should be sort of similar length and should be not too bendable. And then we define an image energy that tries to match where the pixels are the darkest, because that's where the flagellum is. The nice thing about this is that you can sort of do half of that minimization through the Euler-Lagrange equations. And then here's a stupid example where you showed that you can also use it to find contours. That's actually what you typically use these kind of algorithms for. You draw a circle and then minimize this thing looking for contours, and then you can find the perimeter of a face, for instance, but for the flagella, it looks something like this. So here I've just tracked the base, and then you need to do a few other things. You need to track overall body movements and all these things. But then you get flagella tracks, even though the video quality is not great. And the tracks, if you take and plot the final beating angle, the signals look something like that. So for a single beating flagella, you can calculate the autocorrelation that's shown up here. And so it shows that, okay, it does keep sort of a constant frequency, but it shifts a bit in phase because it decays on sort of a time scale of 0.2 seconds. So even though the beat is fairly constant, it does decorrelate with itself on quite short timescales. And more interesting for me is that there was none of the track flagella that I found had any form of synchronization. A very, very tiny signal can be made out, but I think that can be attributed to just the overall colony moving a bit. So here's an organism that forms colonies but doesn't have any kind of synchronization. Yeah, and you can also see that there's a broad distribution of frequencies. This decay time has a broad distribution, but no cross-correlation.
Here's a question, I have a question.
Oh, yeah.
So if they don't synchronize, how do they swim then? I mean, I thought I have heard Ray giving, in the opening workshop, for example, and my understanding was when the flagella synchronize, that kind of helps the sort of like.
It definitely helps, but you don't need to be synchronized to. I mean, it depends. In Valvox, I think the metrochronal waves are quite important. But I mean, you could imagine two cells sitting like this, beating, but being out of, like not being synchronized in any way. You would still swim that way. I mean, there's nothing preventing that. But maybe if they were synchronized, they might add up to a bigger propulsion. Could be, but.
But what? Okay, so essentially, what I'm asking. You can still have a well-defined velocity from that. It's not that it appears that that
they're not jiggering back.
Yeah, it's not like a diffusion process or something.
No.
At least on the time scale of interest.
No, they're still swimming just fine. Yeah.
Because one flagella by itself works, right?
Yeah, okay. But I was kind of thinking, but could you have like the one canceling the other?
But then they'd have to be sort of synchronized in a bad way, right?
Yes. Okay.
Yeah.
Sorry, and then a question on your next slide.
Uh-huh.
Are those samples in these histograms, are those different flagella? Are they different choanoflagellates?
Different flagella, yeah.
Okay.
Yeah. But of course, all cross correlations are within the same colonies, right? They're different flagella, but within the same colonies.
Right, that's for the last figure.
That's for the last.
But for the tau and the F, you're just sampling a bunch of.
Yes, exactly. Yeah. Okay. So now I sort of want to go over and talk a bit about random walks of these colonies, or colonies in general. So this should be very, very familiar to you. Random walks, you end up getting squared. R displacement goes linear with time. Very well known. Also well known now, but maybe it wasn't 20 years ago. I don't know when. Is that of course you can also have random walks, even though you're swimming with a constant velocity, you can still do random walks because you're also going to have fluctuations in the angle in which you're swimming. So if you swim with a constant velocity but have rotational diffusion, what you end up seeing is that you have a ballistic timescale early and then turning into diffusive later. So let me just see. So I'm sure many of you have done this calculation where you can calculate the square displacement for such a swimmer with a constant speed. And what you end up finding is an effective diffusion constant. That's the translational diffusion constant plus this active diffusion constant, which goes like speed squared over rotational diffusion. Maybe a bit less well known is that when you go to higher dimensions, obviously the translational diffusion constant doesn't change, but the active diffusion constant does depend on dimension. So this is 2 in 2D. It would be 6 if it was in 3D and you are rotational diffusion around 3 axes.
So you would have a theta and you will have a phi as well, something like that, that they're both. I mean,
Well, yeah, yes, you would have a phi and a theta, but you can't just say that phi and theta evolve by, those two cannot evolve in the same way, right? In phi, you will still have the same kind of process, but in theta, you have to be careful because of,
so you mean, what I'm basically asking, then I would have a system of x, theta, and phi, and then in 3D, and is theta and phi satisfying Brownian motion, pure Brownian motion, or
no, because then you wouldn't have proper.
Brownian motion on the sphere, right?
Yes, exactly. Yeah.
Okay, perfect.
Right. So this kind of model is does very well for swimming single cells of S processor, as you would expect. You have a swimming cell moving around, and I think this is an average over 32 cells, and you just get beautiful fits. And it's very nice because it's a quick way to extract both velocity and a effective rotational diffusion constant for these swimming single cells. Right, so here is a sort of fun question that we came up with. So because there were no synchronization, we sort of saw these swimming cells as sort of biological realizations of what happens if you glue together independent random walkers.
We just go back a step. So,
Uh-huh.
There's two D's here. You said you've got a nice fit. So what are the values of the D's?
Yeah. So you want the values?
Yeah, just curious.
So they swim with around, I think, 14 micrometers per second, and DR is around 0.1.
Okay.
Yeah.
So what are they swimming in?
Just water.
Water.
Yeah.
Okay.
Yeah, salt water.
Salt water.
Yeah.
Does it matter how much salt?
No, I don't think so. No, but and there's no way to test that if it matters because they can only survive in salt water.
Okay. So you just have a beaker of seawater or something, or you make the water.
Yeah, it makes salt water. You
put
just
salt in water.
you go buy a normal aquarium salt,
Uh-huh.
mix that with water.
Okay.
Yeah,
Okay.
that's precisely what they live in.
Sorry for the.
No,
silly
by all
questions.
means. And then you add there's some bacteria, so they need some food, so you add a bit of yeast to the bacteria, and that's all they need.
So here you fitted finally a model that has nothing to do with the first part of the talk where you presented hydrodynamics.
No, there's no hydrodynamics.
This is just, so you forget about all of this hydrodynamics.
Yes, yeah.
Take empirical model.
Yeah.
We have two parameters, and then whatever it represents, this model capture about the phenomenology, you are saying that this capture enough, and this is what you can extract with this model.
This is sort of just an effective model saying that, okay, they all have some reasonable constant speed and some noise. And
Although you don't try to relate this to the internal motion, the internal activity of the flagella or anything. You don't care.
No, but so once you have a velocity, it's quite easy to relate that, giving the flagella beat waveform, it's quite easy to relate that to, okay, how much must this beat and stuff.
But for example, how this translates into diffusion, this is not easy. Because you have like a deterministic thing, and ultimately you want to say that this has some kind of
so you would have to make some ad hoc assumptions saying that, okay, the diffusion so there's translational and thermal translational and rotational diffusion, but forget about those are too small. So the beat, you could say it's a constant waveform, and maybe this constant waveform has itself a stochastic term, and as soon as you've given the form of that, the magnitude of that stochastic term can be related to this.
We talked about this. There's noise from the motors that are driving the filaments, right?
So that's for prokaryotes, I think. They're not really motors in eukaryotes. The filaments are active all the way along. So you would sort of have a distribution of activity.
So what's believed to generate the noise in them?
I am not sure.
Okay.
Yeah, but it's very different between eukaryotes and prokaryotes. All right. Well, back to this sort of silly question.
So what happened? when, for example, in the model, you have this, whatever it represents, your particle representing by this equation, hit a wall, how do you reflect, for example?
Well, so in this model there were no walls
There's
or
no
anything, but
Okay.
I mean in principle you could. Well, I don't actually know if that's solved. Is active diffusion solved within a box? I'm not sure, but in principle you could do the simulation of that.
It has
I don't know.
It has a velocity, just reflect the velocity, I guess.
Well, we tried that once,
Uh-huh.
how you reflect a rod. Although it's 2D, it's actually 3D. And it's not anymore, it was not a plane, but it was a surface because of the fact that when you reflect, how do you position the rod?
Oh, you mean if it's not a point particle?
That's right?
Yeah.
So
Yeah, of
I was
course.
wondering if somebody, it's
Yeah.
not that easy.
Yeah, no, but people are doing sort of suspensions of these guys that have hard time, the boundary conditions are not trivial to work
Okay.
out.
No, I wanted to know if you had.
No, I have not touched
All right,
that.
okay.
Yes. Right. So this was sort of motivated by the fact that when you work with stochastic processes, you end up adding them all the time and doing these convolutions. So we were thinking, okay, is it just as simple with random walkers when you glue them together? Do you just have simple addition rules as you have with stochastic processes? So in the beginning, you try to write down just a simple model. So sort of the velocity is the addition of a lot of forces, but you also have but if the forces are sitting and not beating on center, that is, if they're a bit off center, then they'll also induce a torque or an effective torque, because everything is force and torque free here, but effective torque. And so you get this big system. So assuming that these things add up to Gaussian processes.
So, sorry, so just
Yeah.
one thing you just said. So what do you mean by the torque is 0, but there is an effective torque?
He was asking for that, wasn't he?
I kind of was, yeah. So what I mean by that is that, okay, let me just say it with force, just because it's, yeah, whatever, it's the same. So because you're at Reynolds number 0, if you're neutrally buoyant, you must be force and torque free.
If you have no external coupling, right?
Yes, exactly. So what I mean by effective torque and force is that the force that basically the force and torque without the drag induced from the fluid, because they precisely, so the force you act on a particle with some force, that's going to be precisely cancelled by the drag. So what I mean by effective torque is sort of just
Okay.
that
Got it.
force, and then you can relate that linear to the velocity. So it's not like a normal force that gives an acceleration; it gives a velocity. Yeah. So it probably should be defined better than that, but I should just avoid
I
saying
didn't
force and
appreciate that
torque.
point before, but.
Um,
Sorry, did everyone was okay with this? I mean, we like seeing equations, so did everyone absorb the equations in the previous slide, or should we get more?
well, this is sort of the important one anyway.
Okay.
I think.
So those are the resultant. You have many swimmers together.
Yeah, stuck together.
And this is the resultant velocity, resultant moment of all of these together.
Yeah, so you add up all the forces and all the torques, and that's going to give you your initial velocity vector in some direction and initial rotational velocity in some direction. And so that's going to evolve according to these equations. So once you've added up all of this, and the big assumption here is that you still end up with Gaussian noises, but otherwise it's very hard to deal with anything.
So this you assume or this you proved?
That it's Gaussian.
Yeah, I mean the sum of all of this results to this. You assume that you
So
have a large number
okay,
of
so all I'm
limits?
saying is that
Because obviously you have some nonlinearities in the equations, no?
Well, so I mean this adds up to sort of V0 or something, and this thing is also independent of V and X, right? I mean, this is just the positions of the R's are just the positions of the cells. So all of this does add up to something, but whether or not so since this F has some noise and there's some noise there, it's not necessarily that there's going to come Gaussian noise out of it, but it is going to add up to something which might be non-Gaussian, but then some argument about central limit theorem, and I think it's all right to assume Gaussian now.
So the capital omega is like an axis, and the little omega is the rate at which that axis is changing. No, sorry, omega
no,
itself.
the other way around. Yeah, exactly. So basically, you have something swimming. That's going to be rotated by delta big omega, but it's also constantly rotating around another axis, and that you also have to rotate by delta omega. So the final equation looks something like this, and it's important that these cross products, they are cross products, but that they are Stochastic, because otherwise this diverges to infinity.
But Stochastic is only for equation 37, yeah?
No, 25 too.
But there's no dw there, strictly speaking, so.
No, the d omega is.
The d omega has a dw.
Okay.
Yeah.
And why, I mean, and you get, I mean, the Stochastic, do you get that, or is this a model choice?
Well, so I mean, I mean, in some sense it's a choice, but it's the only choice that works. Because if you choose Ito, you're gonna have the velocity and rotational velocity go to infinity. So basically, what's going on is that so the reason this works
So you want V to have its norm preserved, right?
exactly.
Okay.
Yeah, and it works with the normal cross product that because it's constantly acting.
While data will give you some sort of like an effect.
Exactly. Yes. Thank you. You said it better than I did.
You're still responding to him because I have a separate.
No, please.
The DWs in 26 and 27 really are supposed to be the same noise, the same realization of Brownian motion?
Yes. Otherwise, it's not proper.
And so essentially what's going on is you have sort of, there's noisy motion, what's going on? So it's like there's noisy motion around an axis and noisy rotation of the axis of rotation?
Yeah, precisely. Yeah.
And some other both rotations, so they should be the same kind of.
I mean, you want to rotate. You want to rotate the axis that the organism is rotating about. You want to rotate that the same amount as the particle is rotating.
Okay.
There's a lot of rotation words, but yeah. So Stochastic is not very nice for computational simulations. So you can also take this. So what I meant by the Stochastic cross product is this matrix onto that vector. So you can calculate the Ito version, and then you end up getting these noise-induced drifts that precisely make sure that the norm of the velocity and the rotational velocity are conserved. Okay, so this model is unfortunately too hard for me to solve anyway, so I'm gonna make probably horrible assumptions, but maybe some of you can fix that later.
This is not trivial because I mean, I think it's linear. There's a non-commuting matrix multiplication.
Yeah, and I mean the big issue for me at least I thought was that these two have to be the same.
Oh, that's not that.
Yeah, well,
But in principle, you would be interested to have an exact solution for that.
I mean, that would be amazing, but
Okay.
the approach that I took was, okay, first of all, let's just consider the case without noise. Then it's very easy to simple, and you get the analytical solution for helix. If you write that solution in this way, where you define these effective velocities, and R is some orthogonal matrix. That's one way to write the helix solution.
So what is the physical meaning of omega naught?
Omega naught. That's the rate at which the colony is rotating around itself.
Just deterministically.
Yes. So so right now I've taken the noise away. So yes. I mean now it's just going to be.
You would expect there to be so there is some sort of.
I mean there's going to be a mean rotational rate, and that's omega naught. Right. And R is some orthogonal matrix defining the direction of this helix. And so one thing.
Sorry, one more thing. I understand how omega naught as a scalar you would have make sense as a mean, but is there a mean vector omega naught?
that probably doesn't make sense.
Are you referring, I'm just asking because I see omega naught, that V naught, all this type of thing.
Uh-huh.
I definitely can understand there being scalars V naughts and omega naughts that are properties of the organism.
Scalars V.
Oh,
Yeah, okay, yeah.
Well, I guess that's the point. You're saying that there might be some fixed relationship between how V naught and omega naught are related to each other, even though as the thing rotates and moves around, of course
That's precisely it, right? Because they're being rotated, the means ones are being rotated by the same stochastic vector.
So they're both locked into body axes. right?
Exactly, yeah. Yes. Thanks. So this R, one thing that you could do is to say that, okay, when dr is larger than 0, then at least for early times, this is going to hold. It's going to hold that the orthogonal matrix can be written as a multiplication of 3 orthogonal rotation matrices. It's not going to hold for late times, but it is going to hold for early times. And so it might give us a good early time solution. And we are interested actually only in early time, because when you calculate these squared displacement, you really don't care about what's going on after 5 seconds or something. You care about the early time where ballistic becomes diffusion. And as soon as you have the linear part, you have most of it. And after that, it's just going to stay linear somehow. So we are interested in an early time solution. And since this is valid at least at t equals 0, it might give us a good early time solution. And so doing that, you can actually solve the system. And it becomes quite long equations, but you do solve it. And you find that the effective diffusion constant splits up into terms that look like the ones for a normal single-celled random walker that's proportional to these velocities squared. But you have one of them that looks very much the same, and then you have one term where the magnitude of this also decays with the rotation rate. So let's see. So here's a video of tons of these rosette colonies swimming around. It's a bit too low frame rate to make any sense of it. But one big problem with this is that if you take a lot of these colonies and average over them and plot the squared displacement, you're also going to average out the difference in colony morphologies between the different colonies. So you really don't want to do statistics like you don't want to do ensemble statistics, you want to do single colony statistics. So here are 3 example tracks, long tracks of single colonies swimming for around 25 minutes. And so what you see is that indeed they do like sort of qualitatively look like noisy helices. So here's a zoom of the blue one, and you can sort of see the helix structure, but there's a lot of added noise.
This is due to the projection because you're doing 3D? Or
Yeah,
this is
yeah. So I'm not tracking in 3D, but they are swimming in 3D.
It's 3D, okay.
Yeah. So if you do the squared displacement of these kind of swimmers, you have again early time ballistic. You have late time going into diffusive, but in the middle you have these weird bumps that you don't usually see in random walkers, and on this side is the autocorrelation of the velocity that oscillates with precisely what I called omega naught earlier. So if you try to use the early time solution of the model, it actually gives very, very nice fits. So just as in the unicellular version, this allows us to quickly, just from a trajectory, extract the swimming velocities both in the swimming direction and along the helix circle, sort of, and also the noise and rotation rate all simultaneously. So that works quite well.
So that's each line is one track.
Yes. Yeah. So these 3 colors corresponded to these tracks.
I bet not all your tracks work so nicely.
Huh?
I bet not all your tracks work so nicely.
All of them. I mean, as long as you have tracks that are this long, they will work that nice.
Why wouldn't you?
Huh?
Why wouldn't you?
Because it's hard to keep track of the same guy for 25 minutes. Because they're swimming around and you have to actually.
Software issue.
Well, so first of all, if they were swimming in, if I could confine them to precisely 2D, then tracking them automatically would be fine. Like I have software that does that, but tracking them in 3D, you also need focus. And whereas it's very easy in an image to see that it swam right or it swam left or something, it's very hard to see when it goes out of focus if it swam up or down. So you can sometimes exploit that the point spread function of the.
So your imaging is not stacked. It's not 3D imaging.
No, that's not. Huh?
Is it confocal or?
It's normal bright field. But even confocal would be hard because when they swim out of focus, it's hard to tell if they swim out of focus up or down.
Yeah.
And so you'll have to adjust the focus looking for it, and then sometimes they're just lost.
Okay.
So getting these long tracks, it's hard.
Are you the one doing all this?
Yeah, I have a collaborator, Alan, who also did some of these tracks. But yeah, I wrote all the software and stuff, and then we took turns doing the tracks.
I mean, there's the worry that like some tracks are harder to find than others, so you're biasing any statistics that you have.
If I was going to do a very big data set statistics, yes. So I'm actually going to talk about this in a second because we didn't get that many long tracks, and here are the tracks that we did get, and sort of plots of the different fit parameters, and they're just all over the place. And I'm not going to try and do statistics on this because it's first of all, yes, it might be biased. Second of all, it's a very small data set. But nonetheless, we were curious whether or not there was something that could be said about bigger colonies swim faster, or at least colonies of the same size behave similarly, or something like that. That was a question that we had. So instead, we tried to do this kind of things on just very short tracks. So not trying to get all the parameters out, but at least just get speed up as a function of area. So we would fit ellipses to these swimming colonies. And so here, the ellipses turn green when they're in focus, and then we use the area of that ellipsis when it's in focus as an estimator of the size of the colony. So in this way, you can get very, very high statistics very quickly.
And what's the typical range of numbers? This is 100,000
Of what, sorry?
of the colony. How many bacteria you have in this spot?
Oh, how many cells?
Yes, how many
Yeah.
cells?
It varies from 1 to, I think the largest we have is, you have to count by hand to get it exact,
Yeah,
but it's around 25 max.
25.
Yeah.
So when you, okay, all right. And so you did this, so what you're saying is ultimately the large number limits where you went, we account well for things that go from an ensemble that has ultimately a small number of particles into it, like say 10, it can work very well already. When you do this approximation of the equations, and all right, okay.
Yeah, if I understood correctly. So that kind of statistics leads to another mess, or whatever you want to call it. So all of the blue circles are the speeds of these swimming colonies. The green circles are from the long tracks. So luckily they have the same kind of distribution to them. But what it does show is that even two colonies of the same size can be very, very different. So what this says is that the formation of these colonies, they form by cell division. So the cell divides, and then you end up sitting at some orientation. And so the fact that you can be the same size but swim very differently says that this colony formation is very stochastic itself. So I've not gone into detail with this, but just stating the fact that even the colony itself must be quite stochastic.
If
So
you forget about the flagella, is the shape stochastic as well? Just a pure geometric outline?
they all look very spherical, so it's hard to tell, and it's very hard to see the flagella, especially what direction they're pointing in. But no, the shape itself does not look. That's
You know, you don't get like elongated crescents or anything.
the only thing that you do get are dumbbells sometimes. It looks as if one cell has divided over here, and then a second colony has started, but they're swimming together like this. So you have these dumbbells, and they typically rotate very quickly around themselves.
On your star.
Right, so the only surprising fact I think here is that we don't have many slow swimmers. So when you think about it, if you're sitting in a sphere, you would tend to cancel out your forces. So I would have expected that the larger you are, the more you're canceling sort of, it should sort of, the swimming speed in my like a priori opinion should decay with 1 over square root n or something, but it's actually not doing that, which I haven't got a good
You might
explanation
be
for.
less likely to see those in your field of vision.
That is a good point, yeah, yeah.
How long do the cells live?
Around, I think 30 hours or something.
Okay,
Yeah.
and would they normally divide before 30 hours? Or
Yeah, yeah, yeah. The life cycle is around. Yeah, so they will, yeah, they'll typically. Wait a second. No, sorry. They'll divide, and then to form a colony takes about 20 hours, and then
Okay.
that lives on for
So
around.
each
30.
colony comes from one cell originally?
Yes, that divides,
Okay.
stays together, and then sometimes they'll spit out a single cell that will make a new colony.
Okay. And so these colonies of 25 cells,
Mm-hmm.
they came from one cell in 24 hours?
Yeah.
So you've had, okay,
Few
3 or 4 rounds of
divisions.
division.
Yeah, yeah,
Okay.
yeah. Yeah.
So I don't know if you did this, but when you showed those like histograms of the frequencies and other properties of different flagella, if you were to make histograms from a single colony, would they tend to kind of be more focused in one part or the other, or would they also be kind of broadly distributed?
So, sorry.
So like, for example, you had a histogram that might have had 30 samples, different, some lower frequencies than others.
Yeah.
If I just looked at, say, the 5 flagella in a given colony,
Mm-hmm.
would they also tend to kind of be distributed similarly, or
Yes,
because they came,
yeah. It's so even though they share the same mother cell, they'll
Yep.
still be very
Okay.
different. Yeah, exactly.
And do you have a histogram of the distribution of sizes?
No, but it's, I mean, in terms of area, it's very easy to obtain from
It would be
this
nice.
plot, right? I mean, it's basically you can draw that with your eyes almost. But, I mean, then again,
I mean,
that's under the assumption that it's not biased sample, but.
Yeah, so all the size are I would have thought, that big size would have been.
Dominant?
No, on the contrary,
Oh.
it's very hard to make big sizes while it's very.
I mean, you
because
can also
it
see
can
that
say-
it is sort of hard to get at least very big.
It would be interesting to understand the process of aggregation and
There's no
dissociation.
aggregation, it's
When
cell
they-
division and they stay together.
And they stay together.
Yeah.
They never.
No.
Never.
No.
Oh, okay, I see. Okay.
Yeah.
Do you have a similar thing for the omega 0
Uh,
as a function of size? Similar scatter plot?
So first of all, omega 0 could be estimated so when you look at the video, this, for some of these swimmers, it could be estimated, but not for all of them. So there's some bias already there when I'm saying that for some of them I could estimate omega 0, and that's typically the ones where the trajectory varies a lot. But for those, surprisingly as well, I didn't find that it decayed with the size of the colony as it did in your paper on the colonies induced by that polymer. I can't remember precisely what it was.
Yeah.
Um,
The E. coli depletion.
exactly, yeah. So we didn't find that the rotation rate decayed with size at all. I have the plot somewhere, but I'm not sure it's on this computer.
So it's somehow deceptive because everything's in 3D, right? But
Yes.
It looks 2D.
Okay, so sorry. So this
Maybe
movie
things
was
are
actually
very,
in pseudo 2D.
Okay.
Yeah. I mean, no, it is 3D, but it's in a very narrow gap.
Uh-huh.
But the previous tracks were full 3D.
Okay.
Right, so in the last section, I'll talk about aerotaxis, which is something I noticed, because in some of the videos I had very high concentrations of choanoflagellates, and then sometimes you would accidentally have an air bubble trapped between two cover slips. And what I found was that if I had high concentrations, after a long time, they would suddenly concentrate around the air bubble. So that could say something about them wanting to get close to oxygen. So it could also say something else. It could be something along, there could be some surface tension explanation or something. We weren't quite sure. So we ended up doing, oh, there's a, okay, I'll tell you what we ended up doing in a second. But first I want to just quickly talk about Cladomonas. Because Because Chlamydomonas is a great example of swimming up a gradient of something. They're algae and they like to find light. They have this eye spot here, and then when they swim, they also rotate around themselves. And then I have sort of a light there, and I rotate around myself. And then when I see the light, I have sort of a timed frequency by which I enhance what I do with one flagella and decrease what I'm doing with the other flagella, and I rotate and do the opposite. And that makes the Chlamydomonas navigate towards light. It's a very nice mechanism, but it only works because the internal chemistry that sets the time lag to increase or decrease the flagella beat is timed with the rotation rate of the cell itself. Otherwise that wouldn't work. So already here we can say that because we know these colonies, they rotate at a stochastic rate, they cannot have internal chemistry that's timed with their rotation rate. Because all the different colonies, because they have stochastic morphologies, there's no way of them knowing how fast they rotate around themselves.
You're talking about the choanoflagellates.
Huh?
The choanoflagellates are that's what you're referring to now?
The colonies of choanoflagellates, yes. There's no way that they can have internal chemistry timing with their rotation rates as these do.
I was getting confused. So these things, are eukaryotic?
Yeah, they're plant cells, algae. Right, so to test this, built a microfluidic device. So here is a sketch of it. The idea is sort of the purple chamber in the middle is where the choanoflagellates are going to live. And so when we add choanoflagellates here, that's here, you pop them in like that, then they are in that middle chamber, and then you build this chamber in material called PDMS. And the great thing about PDMS is that it's permeable to gases, so you can have gas running in channels surrounding the main chamber. And then, so if you run oxygen in one gas channel and nitrogen in another gas channel, you're going to have a gradient of oxygen build up across the sample chamber. And so hopefully that will lead to them swimming towards oxygen.
What is the width here? I mean, an idea of the size? I mean, it's micrometer, tens of micrometer, millimeter.
So this is around 100 micrometers tall. That chamber is around 4 millimeters tall. Yeah. Sorry, and this is how it looks under the microscope. You have the glass, the chamber sitting on the microscope with all these channels coming in and flowing gas. And indeed, we do find that the colonies, they end up all staying at the oxygen-rich side. So that's very nice. So here's just a quick simulation of the gas distribution in the chamber. So you can sort of imagine that this chamber you saw before, make a slide through it, and then you can simulate the diffusion equation, and you get the distribution of oxygen throughout the chamber. And this can be used very nicely to color code videos of these guys. So now there's oxygen at the top, but then I'm gonna swap which channel flows oxygen and which flows nitrogen. Now the simulation is down here, and then you can see that they very nicely and quickly migrate towards the other side. And it's a bit sped up. You can see the time scale at the top. After a while, they do end up down there. It'll take a while before they're all, some are better than others. Okay, one other thing you can do is that you can have nitrogen flowing on both sides, and then suddenly change that to nitrogen on both sides, and then you're going to squeeze them into the middle. So here we can actually keep them in the middle part of the chamber by having nitrogen flowing on both sides. So
They
they'll
don't
still,
like nitrogen.
huh?
They go away.
Well, so there's still some residual oxygen left in the middle, and they'll stay there. So this kind of experiment, when you switch on oxygen, they, of course, quickly go out there. All right.
Is it known that this kind of is it like a chemotaxi where they sense the molecule of oxygen and so they're flying through the gradient? This is what happens?
I mean, I mean, it must be something like that.
Is it known or this is, yes, sir. Take your time. Just
Oh God.
sorry.
Please ask.
Let's
I mean, so first of all, there's no chemotaxis, any taxis known for choanoflagellates before this. So this is basically the first one for
okay.
choanoflagellates. But yes, it's very similar to chemotaxis. They must somehow be sensing oxygen. Oh, my voice is still. Oh.
So now David can ask more things. You see.
Yeah, somehow they must be sensing oxygen and somehow biasing their walk. It's going. It's completely wrong, my voice now. Sorry.
It would be very interesting to look at these trajectories. Have you analyzed the trajectory when you start with the oxygen to see what you can analyze?
So I'll show you what I've done, and then I can tell you what I wish I could do.
Okay.
So first of all, you can plot the density evolution of that kind of experiment that I just showed you. So somehow you can estimate the density of choanoflagellate colonies all over the place, and you get these kind of plots as a function of time, heat maps of where the choanoflagellates are. And so when you have nitrogen and oxygen on opposing sides, you see the obvious there at the oxygen side, and you also get the drift velocity of the population as a whole, which turns out to be 1/3 of the swimming velocity. So they're quite good at biasing their walk. But very interesting, I think, is that here you can see that if you have oxygen on both sides, you have them sort of ending up in a uniform distribution. But if you have nitrogen on both sides, you have them staying in the middle. So there's sort of an asymmetry. Like if this had been symmetric, you would expect them to stay at the side in the oxygen everywhere. Yeah?
But I mean, it's not a surprise if you can explain again what you said, because I mean, in some sense, looking on each of the two figures individually, it's not a surprise. Yeah, in the one, there is more or less oxygen everywhere,
Yeah.
so they just spread.
Exactly.
And in the other one, there is some, if I understood correctly, there is some oxygen left.
Okay, so my point is just the mathematical point that the residual nitrogen in the middle here will be the same order of magnitude as the residual oxygen.
Okay,
Do you understand what I mean?
and I think so.
So you're saying, yes, there is oxygen everywhere here, but only in the same way as there's nitrogen everywhere here.
They're not sort of repelled by nitrogen, it's simply,
It's simply there's some residual oxygen in the middle here. There's also residual nitrogen in the middle there, but they don't really care about that.
So, but isn't that just a manifestation that they actually really want to be next to oxygen and they don't necessarily care that much about nitrogen?
I think the right way to put it is that if there is enough oxygen available, they won't look for small changes. But if there's absolutely no oxygen around, they'll find the maximum. Yeah. But there's another explanation for you in a second. So the typical descriptions of these types of density evolutions are the Keller-Siegel equation that looks something like this, where you have an effective diffusion and you have an effective advection. And so if you had a constant situation, you would just have a constant drift velocity, but you can also have that this drift velocity comes from a gradient of oxygen. The problem with this thing is that you have you can have with high gradients you can have the drift velocity being higher than the swimming velocity. So a typical fix is to say that okay, you react but you react to the gradient, but you cannot swim faster than some given drift velocity. Yeah.
Can I ask one silly thing? So, but don't you need an equation for C? So, don't you need an equation for C as well?
For C as well, you definitely do. If we're assuming that consumption of oxygen doesn't matter, the equation for C is just a diffusion equation that I have already solved. I showed you before.
Okay.
Sorry, but then the oxygen stays for more or less the same. So you might have actually a Laplace equation instead.
Oh, what, sorry?
Equation. So because the concentration of oxygen stays, this is dependent of time, I guess, right? I mean,
Well,
stays more or less constant.
in the
along
experiment, I have time varying boundary conditions. So if you sort of want to compare this to the experiment, you need to input the solution of the diffusion equation with the boundary conditions of the gas channels in the experiment. So the C does not stay constant in time. The C is precisely this, right? Then I have oxygen everywhere, and then sometime, and then you have these things.
I guess the question is, how quickly does the concentration relax when you make one of these changes?
So how this-
Yeah, so so here we can see how quickly the diffusion, how long time it takes for the diffusion to be stable, and then.
Oh, I see, there, that's the chemistry.
And then we have the density here is sort of same time scale as the diffusion to become stable. And then we have this choice, might seem like a good choice. But in fact, if you solve with this choice, you won't get that asymmetry that I just talked about. But one very natural explanation is to say that maybe they don't send gradients in oxygen, they might send gradients in the logarithm of oxygen, such as many other biological sensors, they sense logarithms like your ears or eyes or something. So if you take the logarithm of concentration, you end up with a similar equation that's slightly different. And then you can solve those. So at the top you have the experiment again. The middle one is solved with swimming in concentration of oxygen, and here concentration of logarithm of oxygen. Logarithm of concentration of oxygen. And we can see that the middle part is quite good everywhere except for the nitrogen section. In the one with logarithmic of concentration, you can actually get this strip in the middle as well, although it's not quite as well defined as in the experiment it is there. So this is sort of a nice way that you can just, from the Kirkegaard equations, you can match the experiment quite well. And that gives you the drift velocity and gives you all these things. But it doesn't answer the question, and that's coming back to what I was just asked, is what's the mechanism that gives this drift velocity? Because, okay, so I can describe this process on a population level, but can I also say something about the mechanism that gives rise to the drift velocity? So one obvious answer would be that they're just biasing their swimming speed. So if I go up a gradient and then I swim faster when I go up the gradient, then I will bias and end up at the site with high concentration. So if you plot this population swimming speed in green, you can see that it actually stays quite constant. And it definitely doesn't change enough to give a drift velocity of 1/3. In fact, you can calculate that if you want a drift velocity of 1/3, then you need the swimming speed. If it were to vary linearly with the gradient, then you would actually have to have the swimming speed become absolute zero when swimming the wrong way and double in the other way. Otherwise, you can't get a drift velocity of 1/3. So that's sort of a hand-wavy argument that this is way too little reaction in speed. So the mechanism must be something else. The very famous example of biasing a random walk is the run and tumble of certain bacteria. So what they do is that they have all these flagella that are left-handed helices, I think. And then when they rotate in one direction, they bundle up into a massive flagellum bundle, and they swim in one direction. But when they spin these flagella the other way, the flagella point out in all different directions, and they tumble and choose another direction. So by biasing how often it does these tumbles, you end up with bacteria doing chemotaxis. So I don't see any tumbles in the aerotaxis data, but you can still come up with sort of two very different types of strategies for swimming. So this, in 2D, say that you want to swim up, you want to go up. So there's two ways. In the top line, I've said that the the red is sort of a deterministic change in direction towards the right direction with some added noise in blue. That's what I would call a deterministic strategy to swimming towards some chemoattractant. And in the bottom line, you have a stochastic push, but the size of this stochastic push is biased by whether or not you're swimming in the right direction. So those are sort of two very fundamentally different ways of swimming. And from a mathematical point of view, I feel that these are very simple equations, like describing these two kind of strategies. So the one here, you have a deterministic change with some added noise, or you just have a noise term, but the size of that varies with which direction you're swimming in. So what I actually would have liked to do, of course, is add chemotaxis to the original equations that I had for my colonies. But that turned out to be quite difficult. So as a sort of first approach, this is what I've done. I've just said, okay, let's consider this a 2D swimmer and try to do statistics with these kinds of models. So the nice thing is that you can solve them both for the steady state case and for the distribution of a push or change in direction. And whereby change in direction, I mean this delta phi is positive if you are now swimming more in the right direction and negative if you're going away from the right direction. And so trying to do this on the on the data, what we find is that the deterministic model, the blue one, cannot fit the data. So in order to have this final, when you're swimming to the other side, you're going to have some distribution of angles. Some are swimming in the right direction, some are swimming in the wrong direction, and the distribution looks like that. And in order to fit that with the deterministic model, it actually has to shift the push to the positive side. But the data says that it should be around 0. So just with these effective models, we can actually say that the way it biases its motion is by doing adding some kind of biased stochasticity to its swimming. But it would be very nice if these kind of chemotactic changes could have been added to the full sets of equations. I think that was all I had, which I'm also a bit over time, so that's good.
We started late.
Oh yeah, we started late, that helps. Collaborators, Raymond Goldstein, of course, Kyriakos, who's sitting in the corner, and then Alan and Amber. Yes, thank you very much.
Any questions?
We had a lot.
So I think maybe if there's no immediate questions, we'll thank Julius again, and if people are interested in, are you still okay to chat for a bit?
Sure.
Okay, so then we'll just have sort of a formal conversation over coffee afterwards.